Chapter 21 is miscellaneous bacterial agents of disease. Okay. So pathogens that are not fitting the categories of gram-positive or gram-negative, we talked about in the last 2 times. Gram-positive, gram-negative rods or cocci include spirochetes, curviform bacteria, and obligate intracellular parasites and mycoplasmas. That's what we're going to be covering today. This gives you an idea of some of the organisms involved, what they affect, whether they affect the skin, the epithelial system, the nervous system and muscles, cardiovascular, lymphatic system, gastrointestinal, respiratory, or urogenital tract. Thanks to some of the different bacteria affect different systems. We're going to go over each one of these, whether it's Lyme disease, leptospirosis, gastroenteritis, things like that. And we'll go over each of those, the different bacteria that affect those. And you also got cat scratch fever, or Rocky Mountain spotted fever. One type of typhus is epidemic typhus. All right, we're going to go over all these different types of bacteria and how they affect us, kind of like a summary. So the spirochetes, okay, they are thin, regular coiled cells. Okay. They live in the oral cavity, your intestinal tract, the perigenital regions of humans and animals. Okay. Pathogens are strict parasites with complex growth requirements. They require live cells for cultivation. They have to live inside a cell. At each end is the flagellum, an endoflagellum. Okay, the cell body is, you see the layer inside here, this is your cell body. The periplasmic space is a place between the outer membrane and the cell body. Okay, that's what they look like. So the genus Treponema are thin, regular coiled cells, just like we saw in the picture. They live in, okay, just like we talked about, okay? They're the same requirements that we just talked about. This is the genus Treponema. Treponema pallidum is what causes syphilis, the STD. Human is a natural host for syphilis. It's extremely fastidious and sensitive, cannot survive long outside the host. Okay, it is sexually and transplacental transmission. So it goes through the placenta or through intercourse. Okay. The syphilis disease has several stages. There's an incubation stage, which is the first stage, and that usually lasts around 3 weeks. Okay. During those 3 weeks, there's no lesion. Treponemas adhere and penetrate the epithelium after they multiply. Okay. It's an asymptomatic phase, and there is no treatment because there's no symptoms and no lesions. You don't need to have it. Then you have primary stage after incubation. That occurs between 2 to 6 weeks. Those are the initial appearance of the canker at the inoculation site. It's an intense treponema activity. The virus is multiplying, the bacteria is multiplying tremendously. It's multiplying very quickly. Okay. Active in the body. And then later on, the canker starts to disappear during that duration, during the primary phase. The diagnosis is dark field microscopy, VDRL, VTIB, MHA, and TP testing. Those are the tests we talked about before. Okay, the treatment: Bethazine, penicillin G, 2 million units, and doxycycline, combination of drugs. Then you have primary latency, which is 2 to 8 weeks, so a couple of weeks after that one, extends past. You have a healed canker sore, little scarring, treponemas in blood. There's few, if any, present as it goes. Serological tests are positive now, so your blood tests are positive for it, and the same treatment. Okay. Then you have your secondary stage. This is 2 to 6 weeks after the canker lesion disappears. In clinical, at the skin mucous membrane lesions, you have hair loss. Patient's highly infectious at this point. Okay. Fever, lymphadenopathy, and the symptoms can persist. You have the same diagnostic techniques, dark field microscopy and serological testing, same treatment. And then you have a latency period, which is 6 months, which is lives as a parasite inside the cell, 6 months to 8 or more years. So it can be lay dormant, just like we talk about hepatitis. I'm not hepatitis, I'm sorry, herpes virus. It can lay dormant in your neurological system and cells for years. This can do the same thing. It can be 6 months to 8 or more years. Treponema quiescent, unless relapse occurs, lesions can then reappear. It's seropositive blood tests, and again, same antibiotics. And the last stage is a tertiary stage. So primary, secondary, then tertiary. It's variable up to 20 years. It affects the nervous system, affects the cardiovascular system and the lymphatic system. Gumas develop in organs, and it becomes seropositive. Treponema may be demonstrated by DNA analysis of the tissues. All right, so this is primary syphilis. Spirochete binds to epithelium and starts to multiply. Forms a lesion called a canker at the site of inoculation. It's commonly found in the mouth or in genital areas. Fluid from the canker is very highly contagious. That contains your pathogen. That's responsible inside the cells. Okay? Canker spontaneously heals as the spirochetes move into the blood. Secondary syphilis. Spirochete is multiplying in the bloodstream. A rash forms on the skin, palms and soles with fever, okay? Headache and sore throat. Rash does not hurt or itch and can persist for many months. The rash disappears on its own. No treatment required for the rash to disappear. And that's what it looks like. Those are the signs of secondary syphilis. Is where you get your rash on the hand. In latency, after secondary syphilis, you have latency and then tertiary syphilis, your last phase. After resolution of secondary syphilis, about 30% of infections enter the latent period. That latent period can last up to 20 years. If left untreated, tertiary syphilis is going to form. Okay, damage to multiple tissues and organs with tertiary syphilis, and gumas. Gumas are these tumors that develop. Okay, they're painful, swollen tumors that can be developed. Are gumas. This is a guma. See how it forms and develops there. And you see the pupils. Look at the shape of the pupil in tertiary syphilis. Okay, you see the shape is different. Pupils are usually completely round. You can see how the shape is different. So it affects multiple systems in the body, multiple organs. Congenital syphilis. Okay, Treponema pallidum can also pass through the placenta into the fetus. The infection is possible in any of the 3 trimesters. Okay, symptoms include nasal discharge, skin eruptions, bone deformation, and nervous system abnormalities. We see it a lot in teeth too. You have what's called Hutchinson's incisors. Those are teeth that have instead of your tooth normally looks like you have a lateral incisor, that's your upper incisor. It comes down. I am not the great artist, but you have your root and the root comes down. With a Hutchinson's incisor, it comes down like a barrel and it has a notch inside of it. And usually they're a lot smaller. That's a Hutchinson's incisor. You have a notch in the surface. And that's one of the diagnosis for syphilis with kids that have congenital syphilis. You'll see what's called Hutchinson's incisors. And they're usually very small and they have that notch in the center. Stigmata in the bones, eyes, inner ears, joints, concentration of Hutchinson's incisors. Those are Hutchinson's incisors. You see the notch and how they're smaller teeth. That's diagnostic of Hutchinson's incisors, congenital syphilis. Syphilis diagnosis in testing. Okay, the different stages of syphilis can mimic other diseases. So you have these overlapping symptoms with other STDs, which makes the diagnosis a little tricky sometimes. Consider the symptoms, the medical history, microscopic evaluation, and serological testing. Staining techniques using silver makes spirochetes more visible with bright field microscopy. Okay. Serological testing, detect antibodies presence, 2P pallidum. We know how to do those different tests. And the treatment is penicillin G. That's what they look. Remember the long chain winding spirochete. So if a patient presents with a spotted rash and tests positive with VDRL test for syphilis, the most likely diagnosis is what? Primary, secondary, tertiary, or congenital.
Is it secondary?
What?
Is it secondary?
Secondary. That's when you see the rash. You see the rash in secondary syphilis. Perfect. Okay, non-syphilitic. Treponematosis. Okay. T. pallidum, and this is the gel. Okay. T. pallidum, endomycum, deforming childhood infection of the mouth, nasal cavities, body, and hands. Occurs in the Middle East, North Africa. Yaz. Treponemus pertenu, invasion of the skin, cut, cause a primary ulcer that sees a second crop of lesions. Africa, Asia, South America. And Pinta. Okay. Superficial skin lesions that depigments and scars the skin. Found in valley regions of Latin America. The different types of the pathogens that can be found. You can see the different types of lesions. You can see on the thorax, the abdominal cavity here, and on the legs, and different bone deformations, like with the look at the feet. Okay. Leptospira and leptospirosis. Those are tight, regular individual coils. How it coils around with bends or hooks at one or both ends. There's only 2 species in that genus of pathogens. L. biflexa, which is harmless and a free-living saprobe, and L. interrogans, which causes leptospirosis and is a zoonosis, transmitted from an animal to humans. So in Leptospira interrogans, bacteria are shed in the urine. The infection occurs by contact with contaminated urine. Bacteria targets the kidneys, the liver, the brain, and the eyes. Sudden high fever, chills, headache, muscle aches, conjunctivitis, and vomiting. Long-term infections may result in kidney and liver damage. Okay, 50 to 60 cases a year in the United States. Okay, Borrelia, those are the large spirochetes with 3 to 10 coils, irregularly spaced and loosely connected. Borreliosis are transmitted by the arthropod vector. Okay, a flea, a tick, how it's transmitted. B. hermsii is relapsing fever. B. Burgdorferi is Lyme's disease, what it causes. Okay, so Borrelia hermsii is the relapsing fever. Relapsing fever is wild mammalian reservoirs, killed in squirrels, chipmunks, and it's transmitted by ticks. Okay, that bite the squirrel, get the squirrel's blood and bite and then feed on human and transmit the pathogen that way. After 2 to 15 day incubation period, patients have a high fever, shaking, chills, headache, and fatigue. They have nausea, vomiting, muscle aches, abdominal pain, extensive damage to liver, spleen, heart, kidneys, and cranial nerve. Multiple organ systems are affected. As the parasite changes, the immune system responds, resulting in recurrent relapse. So the parasite is there, you respond to it, you get all the response, then the parasite changes, you get a second response to it. A new antibodies for it. Okay, these are what we call recurrent relapses. Okay, and we treat this with tetracycline. You can see you have multiple effects on it here. This is your body temperature. This is your first antigenic challenge. Then you get a second antigenic challenge as the pathogen changes. Because your initial antibody response reduces the fever. This is the reduction right here. Okay, then reinfection by mutant Borrelia causes a relapse. So the pathogen changes, it mutates. Changes gives you a secondary or another inflammatory response from your immune system. And it changes again, you get the same thing, a fever, and then a spike. This is recurrent. It's called a relapsing fever pattern. And it's because your antibody responds to it a second, third, or fourth time. So in relapsing fever, the primary infection, okay, induces a high fever. So you get an initial antibody response, and that reduces your fever. Then you get reinfection by a mutated form of the Borrelia. Causes a relapse of the fever. Fever spikes again. The immune system reacts to a second antigen from the mutation, and then the symptoms go down again. A new antigenic form where the pathogen mutates, forms a new antigen on the surface. So then you get a high fever spike again and a new set of immune responses of antibodies. That's what about this relapsing fever pattern. Borrelia burgdorferi causes Lyme disease. This is a complex life cycle. It involves mice, deer, and it's transmitted by ticks that feed on the mice and the deer and then transmit it to humans. Okay, so here's your mouse or your deer. The harbor the virus or the pathogen, I'm sorry, bacteria. Okay, so you get the tick feeds on them, you become infected, and it goes back to deer or back to mice, and then it can bite into humans and transmit the disease into humans. Okay, it's not part of its normal cycle because it doesn't go from humans back into the mouse or the deer. So it's called an accidental host. So this is how it cycles through with Lyme disease. Okay. Newly hatched larvae become infected when they feed on small animals like a mouse, harbor the spirochete. The larvae continue development throughout the year. In the second year, the larvae molts into a nymph, which is very aggressive feeding stage. Nymph takes blood from a number of hosts, deer, humans, mice, whatever. On deer, the nymph mature into adult male and female ticks. They mate. The female lay eggs in the plant litter where they hatch and once again begin to cycle over again. Okay. So Lyme disease is acquired by tick bites. That's what causes Lyme disease in humans. It's non-fatal, slowly progressive syndrome that mimics neuromuscular and rheumatic conditions. 57% of people get what's called a bullseye rash. You can't miss it. When a patient walks in, you'll have red. I might as well use red. And you have this rash around it, and it looks like a bullseye. That's exactly what it looks like. You get a fever, headache, stiff neck, dizziness. If untreated, can progress to cardiac and neurological symptoms, polyarthritis. Treatment is tetracycline or amoxicillin. Oh, here's your bullseye rash. And you can't miss it. It looks just like a bullseye. And this is one of the black-legged ticks is responsible for transmission of the pathogen for Lyme disease. And you see the swelling after it feeds. It's just full of blood.
Wow, I've always wanted to see that.
Yeah, it's I mean, 100 times its normal body size. Go ahead.
I was just gonna say I met someone who got Lyme disease.
Huh?
I knew someone who got Lyme disease and said that it affected her like nervous system and she
Yep.
felt she was getting like shocks all over her body.
It does. It affects the nervous system and the joint, arthritis. It's very painful. It can be debilitating too, because it does affect those two places tremendously. But you see what those ticks look like. They're just huge and they're filled with blood. And if a patient walks in with 50 to 70% are going to have this bullseye rash. And I think it's the only disease I know that has a bullseye rash like that. It's very diagnostic. Okay, the curviform bacteria, these are gram-negative. There's 3 groups that share adaptations to survival in the intestines. There's Vibrio, Campylobacter, and Helicobacter. In the Vibrio, this is common-shaped rods. They're a single polar flagellum. Okay, the Campylobacter are short spirals, curved rods, have 1 flagellum. And the Helicobacter are spirochetes with tight spirals and several polar flagellum. Okay. These generally cause enteric diseases in the intestines. This is Vibrio cholera. These are common-shaped bacteria, fermentive and grow in ordinarily recycled media at 37 degrees centigrade, which is body temperature. They possess the O, somatic, and the H flagella, where membrane receptors antigen. Remember, they have the different types of antigens the body responds to. This has the O and the H. Okay? And this is what they look like under the microscope. The epidemiology of cholera is favored by warm, monsoon, alkaline, and saline conditions. Pandemic patterns, they invade the population, cause a pandemic ever since 1961. The L-Tor biotype, this one survives longer as more infectious. Okay. In non-academic and non-endemic areas, can spread by ingestion of contaminated food or water. That's how cholera is spread in areas that it's not endemic to, doesn't grow normally. Infects the mucous barrier of the small intestines, but it's non-invasive. Okay. Cholera toxin causes electrolyte and water loss. Okay. That's when it becomes fatal. The treatment for cholera is antibiotics, but that's not, the main treatment is hydration and electrolyte replacement. And we'll see in a second just how you lose electrolytes with water. Okay. And it causes this through secretory diarrhea, which is called rice water stool. Resulting dehydration leads to muscle, circulatory, and neurological symptoms. Treatment is rehydrate. That's the big one, rehydrate. And then we treat with tetracycline. And rehydration means water and electrolytes. This is normal. This is what happens. This is your intestine. This is your intestines here. As your waste and food and what you drink pass through your intestines, you absorb the chlorine, bicarbonate ion, sodium, potassium, water. All this gets absorbed into the cell, and the cell puts this into the bloodstream where it goes through the body, and your body's happy. Okay. With cholera, it produces the bacteria on one and destroys the lining here. So what happens is this cannot come in, so the water forces itself out of the cell. And when the water forces itself out of the cell, it keeps the potassium, the sodium, and bicarbonate from coming into the cell and going to the bloodstream. So you lose your minerals and you lose your water. They travel with each other. Okay. And this is what causes the diarrhea, severe water loss, and you have to just keep rehydrating the body. Okay. So the pathogenesis, how it affects your cholera, and the toxigenic diarrhea. So the Vibrio cholera cells come to rest in a protected mucus coated near the cell surface and secrete the cholera toxin along the cell surface. The toxin has an affinity for specialized receptors on those cells, on the glycocalyx. The glycocalyx are proteins part of the cell membrane, and it binds to those. The active portion of the toxin gets released, is transported through the membrane and into the cytoplasm of the cell. That's the toxin. So the bacteria are not entering the cell, they're non-invasive. The toxin they produce, after they bind to the glycoproteins there, they bind there, the toxin they produce, those enter the cell. So it's a term non-invasive because the bacteria itself is not entering the cell, but the toxin is. It becomes a signaling system that converts inactive adenylene cyclase into an active state. So this enzyme then converts ATP into something called cyclic AMP. That's what's called, let's shorten as CAMP. It's used in the cells to control a major membrane pump for the negative ions. So it stops that pump that brings ions and water into your cells. The result of this, remember, begins to actively pump chlorine and bicarbonate ion and back into the intestine. Remember, we have to keep chlorine and bicarbonate into our bloodstream. It pumps it out of the cell so it leaves the bloodstream and into the lumen of the intestine. One additional effect of the toxin, it overrides the usual control of the adenocyclase cyclical AMP system. So the cell continues to pump out these ions for extended time. And if you pump out ions back into the lumen of the intestine, you're pumping out water with it. Remember, it goes together, water and sodium, potassium, bicarbonate, chlorine, all those ions go together with water. So where those flow, water flows by osmosis. Okay? So positive ions, sodium, potassium, now follow the anions, which is your bicarbonate and your chlorine, okay? And they're lost into the intestinal fluid, okay? Along with large amounts of water, causing secretory diarrhea and dehydration. Dehydration is what causes the death in cholera. All right, so here's what happens. You can see it here. This is the Vibrio cholera cell. Okay, comes to rest in the cells in the surface and secretes the cholera toxin. So here's your Vibrio cholera, your spirochete, your flagellum attached to it. And you see it secreting the toxin right here. Okay, this binds to the cell membrane, to the glycocalyx proteins, and it binds to that. That's what keeps the bacteria closed. Remember, the key point, which you'll see in your nursing exams, is that the bacteria does not enter cell, the toxins do. Okay. Okay, the toxin has the affinity for specialized receptors like glycocalyx and binds to what we just said. And the active portion of the toxin is released, transported through the membrane, and enters the cytoplasm of the cell. So there's your first 3 steps. Bacteria bind to the glycocalyx of the cell membrane, releases its toxins. The toxins penetrate the cell membrane and enter into the cytoplasm of the cell. Okay. Then becomes a signal that alters the active state of the adenylyl cyclase and affects the cyclic AMP system, okay, which pumps negative ions into the cell. So what happens is these ions here are being pumped out of the cell because it affects the ATP, binds to the cyclic AMP, and the adenylyl cyclase system. So it pumps the negative charged ions out of the cell. The positive ones follow with it, which is your sodium and potassium. And when the ions are pumped out of the cell, water flows with it out of the cell. So you're getting dehydration. Okay, cause a lot of water get into your stool, and you get diarrhea, and you lose tremendous amounts of water and electrolytes. Okay. So other Vibrio pathogens, they're salt-tolerant inhabitants of coastal waters associated with marine invertebrates. Right? So Vibrio parahemolyticus is gastroenteritis from raw seafood, gives you symptoms similar to cholera. Vibrio vulnificus is gastroenteritis from raw oysters. Now, this is a real problem, especially for people with diabetes or liver disease. This can be fatal. Okay, diabetes or liver disease causes a very severe reaction with Vibrio vulnificus. Okay. Treatment is always going to be fluids and electrolyte replacement. Always, always, always. You got to replace that water being and those electrolytes that are lost. Okay. And then you put, if you identify which pathogen is, and antibiotics. So the main symptom of cholera infection, okay, this is the main thing that cholera does. The main symptom of cholera is going to be what? A rash, water diarrhea, open lesions, or paralysis. What's the main problem with cholera?
Watery diarrhea.
Watery diarrhea. We always got to replace that water. That is what cholera does. Causes dehydration from watery diarrhea. Perfect. Okay. The Campylobacter genus are slender, curved, or spiral bacilli. They're often S-shaped or gull-winged pairs. Okay. They have a polar flagellum, common residents of the intestinal and genital urinary tract, and oral cavity of birds and mammals. Most important species in medical and veterinary practices, the Campylobacter jejuni and the Campylobacter fetus. Okay. Now, this is the Campylobacter, what it looks like. Okay. Campylobacter jejuni, remember, the small intestine, okay, of a human is your three divisions. In order, it feeds from your stomach into the duodenum of the small intestine, into the jejunum of the small intestine, into the ileum of the small intestine. Then the ileum feeds into the large intestine. The jejunum is where most of your absorption of all your food occurs. So if you're infecting jejunum, which is Campylobacter jejuni, that's going to prevent you from absorbing a lot of your food. Okay, so this bacteria is going to be transmitted in that area of the small intestine. So it's transmitted by beverages and food, gastroenteritis. It reaches mucosa at the last segment of the small intestine near the colon. That's where it adheres to. It burrows through the mucus and starts to multiply. Starts to multiply. Their heat labile enterotoxin, with CJT, stimulates a secretory diarrhea like cholera. You get headaches, fever, abdominal pain, bloody or watery diarrhea. Okay. Treatment. Get that out of the way. Here you go. It's rehydration and electrolyte balance. This is going to be for any of your nursing exams. Just remember, if one of the symptoms is going to be diarrhea, gastritis, diarrhea, cholera diarrhea, your main treatment is going to be rehydration and electrolyte balance. You've got to restore that. Okay, that's your main goal. Antibiotics, secondary. Antibiotics, erythromycin, azithromycin may be necessary in severe cases. Prevention, there's no vaccine available for it, but Prevention depends on rigid sanitary control of water and milk supplies and care in food preparation. We get it through food and through water. Campylobacter fetus, traditionally to veterinarians. This is a sexually transmitted disease of sheep, cattle, and goats. Okay, its roles in agent of abortion of these animals has considerable economic impact on the livestock industry because you've got to keep that livestock maintained. Okay. In humans, opportunistic pathogens infect debilitated persons or women late in pregnancy. This one is linked to meningitis, pneumonia, arthritis, septicemia in the newborn. This is rare to transmit to humans, but it can. Helicobacter pylori is a gastric pathogen. Those are the curved cells discovered in 1979 in the stomach biopsy specimens. Causes 90% of stomach and duodenal ulcers, apparent cofactors in stomach cancer. They've tried to link that. Duodenal ulcers have a proposed link with stomach cancers, not a definite link yet, but they've proposed a link for several years now. People with type blood O have a 1.5 to 2 times higher rate of ulcers than other blood types. Okay, the Helicobacter pylori produces urease, which converts urea into ammonia, okay, and bicarbonate. This is the Helicobacter pylori. That's what it looks like. You can see the cells, and this is the purple is all the stomach mucosa lining. And you can see the orange is all the Helicobacter cells. Medically important bacteria of unique morphology and biology. So pathogenic bacteria with atypical morphology, physiology, and behavior include rickets, rickettsia, which is intracellular pathogen that rely on an arthropod vector. Again, tick, the flea. Chlamydias, intracellular pathogens that alternate between elementary and reticulate bodies. Now, these are different states the bacteria is in, the pathogen is in, that in one state it cannot become infectious, other state it does. That's what it means, only between these two different states. Both obligate parasitic gram-negative toxobacilli and mycoplasmas. These mycoplasma lack a cell wall and are highly pleomorphic. They adhere to the cell membranes and they form their own shapes, basically, and cause the disease. So rickettsias, okay, are gram-negative cell wall. They're non-motile pleomorphic rods or coccobacilli. They're small, obligate intracellular parasites. Ticks, fleas, or lice are involved in their life cycle. Okay, those are your vectors. Bacteria enter the endothelial cells and cause necrosis of the vascular lining. So you get vasculitis, vascular leakage, and thrombosis being formed. Okay, so these, the rickettsias, these enter into these cells. And that's just going to form the different states too. So these are invasive, and you can see the bacteria entering one of the cells here full of bacteria. They enter the lining. In rickettsia, you have the typhus group. There's 2 types of typhus. Okay, there's epidemic typhus and there's endemic typhus. Epidemic typhus is R. prausnitzii. It's carried by lice. Starts with a high fever, chills, headache, rash. Okay, it has chronic and recurrent form. The endemic typhus is by R. typhi, harbored by mice and rats, occurs spontaneously in areas of high flea infestation, and is milder symptoms than the epidemic type. Spotted fever group. This is responsible for Rocky Mountain spotted fever. This is R. rickettsia. Okay. This is a zoonosis carried by dogs and wood ticks. Most cases are in the southeast and on the eastern seaboard. Distinct spotted rash may damage your heart and central nervous system. Rickettsial pox, okay, is by R. acari, carried by mice bites, and it's found worldwide. Then you get scrub typhus and ehrlichiosis and anaplasmosis. Okay, this is a chart. Well, we talk about the different disease groups that it can cause. So in typhus, there were the 2 types, epidemic and endemic typhus. The vectors are body louse and fleas. Primary reservoir, mode of transmission, Okay, feces rubbed into the bite or inhalation, flea feces rubbed into the skin, inhalation, where they're found. Spotted fever, Rocky Mountain spotted fever by tick, right? And the different types there. And scrub typhus, human ecloses, and human anaplasmosis. And just these are the different species of bacteria that cause them, the disease that results, the vector, how it's transmitted, and where it lives, basically. A mode of transmission, like a tick bite, and where it's found in the world. This is the United States, Rocky Mountain spotted fever. You can see there's 0. If you're afraid of Rocky Mountain spotted fever, move to Nevada, there's 0 cases. Okay, you can see some of the other areas there are 0 cases. The high incidence is in the orange region right here. These are your highest incidents. This is the East Coast and the southern states, where the environment's a lot more moist. Rocky Mountain spotted fever, this is your dog. Tick bites the dog, goes into the human. This typical cycle produces the egg go on the back of the tick goes on the dog, bites the dog, it keeps the infection going. Okay, if the tick bites a human, you can end up with central nervous system damage and eventually coma, the rash, or vascular damage, disease from that. This is a spotted rash. Again, this is Rocky Mountain spotted fever due to the spotted rash that you get. First symptoms are fever, chills, headache, and the spotted rash. Central nervous system can be involved in, and fatality rates are usually 1 in 5 if it's not treated. So the organism causing Rocky Mountain spotted fever is transmitted to humans through a bite of certain ticks, dogs, fleas, or rodents. What was causing the biting here? Ticks. That's the only one that's capitalized too. Must be for a reason. So ticks are the vector for Rocky Mountain spotted fever. If they live on the dog, they multiply on the dog, but the ticks bite the dog, then take that blood and transmit it to the humans. Coxiella burnetii, those are intracellular parasites related to the Rickettsia family. They're harbored by a wide assortment of vertebrates and arthropods. And this is what caused what's called Q fever. Infectious material includes urine, feces, milk, and droplets, usually inhaled, causing pneumonitis, fever, and hepatitis. Tetracycline treatment is what's used to combat it, and the vaccine is available for Q fever. That's what they look like in the lumens here. You can see the bacteria. The Martinelli genus, small gram negative, fastidious, related to the Rickettsia family, considered as emerging pathogens. Martinelli quintana, it causes what's called trench fever, and that's spread by lice. Martinella henselae is an agent called cat scratch disease. Okay, that's lymphatic infection associated with a clawing injury by cats, hence cat scratch disease. More severe infections, bacillary angiomatosis, can result from infections in AIDS patients. Remember, their immune system is depressed, so they're more subject to certain types of diseases. And you can see the types of skin lesions that are found. Okay, the Chlamydia family are small gram-negative obligate intracellular parasites. In its life cycle, often between the 2 stages. We talked about these 2 stages. Remember, we talked about elementary body and reticular body. The elementary body is a small, metabolically inactive. It's extracellular. It's infectious form released by the infected host. So this is the extracellular, is outside of the cell, and that's where it's infectious. And it's not metabolizing. It has to be metabolized inside the cell. The reticular body is the non-infectious part. It's actively dividing and forming more, and it's within the host cell. So the reticular body, so here's your host cell. The reticular body is inside the cell, and it's growing and multiplying, making more. It gets released into these non-metabolics. They're not doing anything, but they are infectious. They'll travel into other cells and infect it. So this is the reticular stage where it's inside the cell multiplying, then it's released outside the cell. These are the elementary bodies. And these are what's infectious. These are ones that infect other cells. In the reticulate form, it's not infectious because it's inside the cell. It can't go anywhere. It can't infect anything else that's inside the cell. The elementary bodies, once it's released, those are the infectious part. Right? And this is what it looks like. So inside the cell, you can see it's multiplying, fission, and keeps growing. And then it kicks out of the cell and becomes these elementary bodies. And these will become infected and they enter into other cells and then start the procedure over again with the reticular body. So that's the difference between those two. The human reservoir, there's two strains in a chlamydia trachomatis, sorry. Two strains can affect humans. Trachoma strain, which attacks the mucous membranes of the eyes, genital urinary tract, and the lungs, and the lymphogranuloma venereum strain, which invades lymphatic tissues of the genitalia. Okay. You can see the ocular trachoma. Severe infection in the eye deforms your eyelid. Okay, and it deforms the eyelid and the cornea and can cause blindness. Inclusions conjunctivitis occurs as babies pass through the birth canal and is prevented by prophylaxis, which is given the mother. If the mother has chlamydial disease, you give them antibiotics prior to the baby passing through the canal, so the bacteria is killed before the baby goes through the canal, birth canal. And again, you see the chlamydial disease of the eyes also.
I thought that was for gonorrhea.
Pardon? Gonorrhea can do the same thing.
Oh, is it part of like the same family?
No, different bacteria. But it can cause both. It can cause blindness and it can cause problems with the eyes.
Okay.
Yep, they both can have those same effects. You're exactly right. Sexually transmitted chlamydia diseases, chlamydiosis, which is the most prevalent STD. It's long-term reproductive damage, asymptomatic in women, 70%, and males, 10%. Non-gonococcal urethritis in males, inflammation of the urethra. It mimics gonorrhea. It's very similar. They do not involve the gonococci. So it's not the same family as gonorrhea, but it can have similar manifestations. Pelvic inflammatory disease in women, cervicitis with a white drainage, endometriosis, and salpingitis. Lymphogranuloma venereum, it's a disfiguring disease of the external genitalia and pelvic lymphatics. And you see the disfiguration in the genital areas. Identification of chlamydiosis. Sampling requires dislodging cells from the mucosal surface. You have a direct assay of specimens using immunofluorescence and PCR-based probes. Okay. GIMSA or iodine stain is used for diagnosing inclusion conjunctivitis. It's not recommended for your genital specimens, too low sensitivity on that test, and you get a lot of false negative results because it doesn't trigger it. That's what that chlamydosis looks like. Beta cells. Chlamydiophilia contains members that used to be members of the genus Chlamydia. So these are two different types. There's Chlamydiophilia pneumoniae and Chlamydiophilia psittaci. The pneumonia one causes atypical pneumonia. Very serious if your patient is asthma. Chlamydiophilia psittaci causes ornithosis, which is caused from an animal vector, transmitted to humans from bird vectors. Highly communicable among all birds, pneumonia or flu-like infections with fever and lung congestion. Cell wall deficit bacteria. This is where your mycoplasmas come in that adhere to your cell membranes. They naturally lack cell walls. They're highly pleomorphic, multiple shapes. Mycoplasma pneumoniae and Mycoplasma genitalium. The pneumoniae is primarily atypical pneumonia. Okay. Spreads slowly over the interior respiratory surfaces, causes fever, chest pain, sore throat. The genitalium, okay, it's a weak sexually transmitted pathogen. Okay, this is the mycoplasma cells and how they attach to the cell membrane, and you can see they got all these different shapes here along where the cell wall would be. So cell wall deficient bacteria, bacteria that have lost their cell walls, so exposure to certain drugs or enzymes can result in cell wall deficient bacteria called L forms or L phase variants. They're induced or occur spontaneously. They may be involved in some chronic diseases. So L phase variants are group A Streptococci, Proteus, and Corynebacterium, Mycobacterium, Avium, paratuberculosis. That's something that can involve this loss of the cell wall. Bacterial and dental disease. The most dental disease of all the teeth and the surrounding supporting structures. The periodontium, which is your gingiva and ligaments, membranes, and bones. Okay. The primary site for dental infections are the grooves in the enamel, like in the cusp, where the cusp come up, they form grooves on there where bacteria can harbor because you can't brush those out. They don't get exposed to the saliva. Right. And the crevice or the sulcus formed when the gingiva meets the tooth. So there's two spots. Here's your teeth. Again, I'm not any kind of great artist. This is your teeth. This is your gums. Okay, and this sulcus right here, where the gums meet the tooth, and these grooves right here. Both areas that bacteria can hide in and not be hit by a toothbrush, not be hit by a saliva, the acid contents of saliva, brushing motion, the washing, something like that. So those are two areas that bacteria can hide. One causes caries, which is cavities. That's this one here, what goes in the sulcus. Another causes periodontal disease, which gets into between the gums and the tooth, the gingiva and the tooth. Okay. So this is your basic tooth, much better picture than I can draw. Again, this is your cusp. Okay, these are the grooves where the bacteria can get into. It also gets into between the teeth, right in here. You can't brush. That's why floss is important. And there's a big thing with flossing that didn't need to be done about a year or 2 years ago, which is totally ridiculous. If you want to prove those studies wrong, just brush your teeth, rinse your mouth, and then floss your teeth and rinse your mouth. If stuff comes out when you floss, it's getting stuff out of there, which is a good thing. So it is important to do that. That's where caries occur. It's between the teeth where food gets stuck and right in these little grooves. And again, here's your sulcus, right in here between the gingiva and the tooth structure. A lot of anaerobacteria, anaerobic, live in here because it can hide from the oxygen content. Okay, they live under the surface there. So the dental pathology, you have the tooth surface is the enamel root, pellicle formation, plaque formation. Okay, plaque is this, the bacteria that adhere to those areas we talked about. Get acidification and cavitation. So once the plaque adheres, those bacteria form colonies there. Once they set their colonies, they feed on sugar and stuff in your food coming in, and they produce an acid. That acid causes acidification of the area, and it causes cavitation, which is a hole into the tooth. That causes your cavities. So they damage the enamel. Tooth destruction, you get exposure of the dentin, which is inside the enamel, and exposure of the pulp and the root. It can cause tooth loss, or cause root canals, things like that. As a plaque, which is what the stuff is called, it's sticky biofilm that adheres to the tooth. This plaque, if it sits there long enough, turns into calculus. Calculus is what they used to call tartar. Okay, it's a very hard thing. We go to the dental hygienist, they scrape your teeth. That's what they're scraping off. They're scraping off the calculus because plaque can get rinsed off with water and toothbrush, but the calculus is not going to go anywhere with a toothbrush. It's not coming off. It has to be scraped up by the hygienist. If it is not, it causes gingivitis and periodontal disease, breakdown of the tissues, bone resorption, lose of the tooth, and tooth loss. The main disease associated with tooth loss in the adult is periodontitis, periodontal disease. So a plaque in dental caries begins with colonization, okay, slim form species Streptococcus, and cross adherence with actinomyces. That's what forms your plaque. Crustus forms a layer of thick adherent material that harbors masses of bacteria, which produce acid and dissolve enamel. Okay, if plaque allows to stay, once it's plaque built up, it sets up a permanent home. Then you get secondary invaders, lactobacillus, bacteroides, fusobacteria, porphyrimonians, and treponema. These build up, they form a thick structure called calculus. Then the acid dissolves the tooth enamel, leading to caries and tooth damage. Okay, so this is exactly what it looks like, how it forms the plaque. So you can see this is your enamel right here. This is your biofilm that starts to form, and this plaque starts to stick. Everything gets stuck on here. You can see it start to adhere to the different layers here. Then it becomes a hardened layer with phosphates and forms your calculus over it. And each step goes through each step here. So a freshly clean tooth surface immediately develops a thin layer of salivary glycoproteins. Where glycoproteins are in the cell membranes. Which you put in your cell membranes. This is your pellicle that starts. This is going to form your plaque. Fibers of the protein, antibodies, salivary enzymes, bacterial debris, and other cellular molecules adhere to this pedicle. Okay, now you've got plaque. The earliest tooth colonizers are the Streptococcus mutans group, Streptococcus aureus, and Streptococcus gordoni. These bacteria have specific receptors that adhere to the outer pellicle molecules. The streptococci likewise bind to each other, forming the initial base for the plaque. This starts to anchor it to the tooth. And this starts within 15 minutes after brushing your teeth. It starts to stick back into it. The next phase involves cell-to-cell signaling and coagulating with additional colonists. The most common bacteria to add to the biofilm during this first phase are filamentous rods of actinomyces. Okay. Other species of Streptococcus mutans, use dietary carbohydrates, that's the sugar that you eat, to secrete glucans that add bulk to the matrix and serve as a source of sugars, food source for bacteria. Once this initial framing has been laid down, it enters a second phase of aggregation, which creates the final dense amount of plaque. Okay, bacteria colonize at this point are frequent the anaerobes, because now you got a protected area where they're not exposed to oxygen anymore. You got a protected where these anaerobes can invade. Okay, Fusobacterium, Polyromanas, Prevotella, Villanella, and Treponema. Okay, initial damage to the enamel occurs when the streptococci near the enamel surface ferment the sugars in the plaque and become to lactic acid, acetic acid, and other active acids. When these acids are trapped against the tooth surface, they etch through it, and dental cavities start to develop. Right. This is the bacteria that are on the tooth surface. And you can see this plaque formation along the tooth surface. That's a scan electron microscope. You can see how the plaque forms around the surface there, and it's not a smooth surface anymore. So microscopic examination of the plaque reveals a rich and varied network of bacteria and their products, along with epithelial cells and fluid. Okay. Now, the periodontal, when plaque becomes calcified in calculus, is because above and below the gingiva. This irritates the gingiva, causing inflammation, gingivitis. That's the definition of gingivitis, inflamed gingiva. So then this pocket where these teeth form, and teeth comes here, comes in here. And as calculus starts to form here, what it's going to do is this is your tooth, and this is your gum like that. It's going to start to create a deeper and deeper pocket. That causes the bone to become lost. It irritates the bone. So pockets between tooth and gingiva are created by bacteria. These are your spirochetes and gram-negative bacilli. The tooth socket may become involved, which is periodontitis. So gingivitis is inflammation of the gums. That's treatable. That's not going to cause tooth loss. Once that forms into calculus and irritates the bone, then you got periodontitis, where you have bone loss. And once the bone gets lost, that's when teeth become infected. So here's the pockets between the teeth. You can see that this is a piece of calculus right here. It causes irritation, and it causes these pockets to expand. And eventually, this irritation continues down, and you lose that bone that supports the tooth, and you have tooth loss. That's periodontal disease. Yeah. So gingivitis, inflammation in the gums. Periodontitis is inflammation in the bone, where the bone actually gets resorbed. And this is what it looks like in a periodontal pocket. You see the different spirochetes, the different bacteria that are found there. Those are a lot of anaerobes. That's why they have that treatment with peroxide. Used to be we call it Kai's technique. It's popular a couple of years back. If you rinse your mouth with hydrogen peroxide, what does peroxide do? It breaks down into water and oxygen, right? Hydrogen peroxide, if you remember your chemistry, goes to water plus oxygen. So if you rinse, oxygen gets released in the mouth, and oxygen kills these anaerobic bacteria. The problem is it can't get way down deep in those pockets. So it's a good idea, but not technically the best thing to do. So a biofilm found on the tooth surface that is made of multiple species is called what? This is a biofilm. So it's flexible. So is it called your caries, calculus, plaque, or periodontitis?
Is it calculus?
Calculus is a hardened one.
Oh, plaque.
That's plaque. After the biofilm forms and stays a while, and then it gets calculus up underneath the gum or above the gum there. So plaque is your biofilm that forms on the tooth.